Hey everyone, welcome to the Microbiology Exam 2 uh, live stream review session. Um, Looks like somebody else isn't here with me. Awesome. So feel free to send me more questions or things you're thinking of or want clarification on in the chat. We can totally use the chat function here. Also, I've got your questions that you submitted in our discussion board on D2L. So I'm going to go through those and we'll just see where it takes us. So I noticed many of you asked about transcription and translation. Great questions. So transcription and translation are the processes that help us get from DNA to a finished protein product. And an analogy I think is really useful with this process of transcription and translation is to think about DNA as a cookbook, and the product that you're trying to make, that protein could be like a chocolate chip cookie. Okay. So if DNA is a cookbook, it has recipes for lots of different things, right? There are genes that code for all the different proteins that it takes to produce a living, functioning, reproducing organism. Just like a cookbook has lots of different recipes to make all the different things that you might like to eat. So if DNA is the cookbook, what is the molecule that functions like a copy of a single recipe? It's a type of RNA. It's mRNA, like messenger RNA. So messenger RNA is a copy of just the chocolate chip cookie recipe. All right. And that copy of a single recipe can leave where the cookbook is stored, and it can go to the kitchen, right? Where we need it. So that mRNA molecule will travel to the kitchen. What's the kitchen in a cell? Where are proteins made? In the ribosome, right? So ribosomes are those teeny tiny little specks that's floating around in the cytoplasm of your bacterial cell models. And those ribosomes are where proteins are made. So when we go from DNA to mRNA, that copy of a recipe or a single gene, that making the mRNA molecule, that's transcription. And then when we go from the mRNA to a growing polypeptide, that's translation. And a trick to help you keep those straight, which comes first, which comes second, you go in alphabetical order. So transcription has a C after the trans part, so it comes first. And then translation has an L after the trans prefix, and so it comes second because L is after C in the alphabet. So transcription is where we go from DNA to mRNA. And when we go from mRNA to a growing polypeptide, that's translation. Yeah. Okay, good. So really it's the going from the mRNA to the tRNA. That's the translation part. Good. So in the ribosome, that mRNA molecule becomes translated into tRNA, and then tRNAs go all over and they gather, they transfer the appropriate amino acids that are coded for by that mRNA codon, and they bring them to the, oh, sorry, I see some text coming in. I'll check at the chat in just a sec. Yes, we can talk about mutations. I'll make a note of that right now, Jen. All right. Okay, so we're going to go from mRNA to tRNA. tRNA goes and gathers the amino acids and brings them back to the ribosome. And when those amino acids get strung together in the proper order, they build a growing polypeptide chain. And when they're added on, that step is called elongation because it's getting longer, the chain of amino acids. Good. Once all of those amino acids are put together, it gets folded up in just the right way, and that is a functional protein or a gene product. Good. So one thing I want to review real quick before we take a step over into mutations is I want to talk about how does it look on paper when we're talking about these different gene products. So what I want to do real quick is I'm just going to open up a blank Word document so I can share my screen with you. All right. So select the tab. Hold on. Change some settings here so it works or not. All right. I don't have administrative access. That's not what I want to do. All right. Let's try a different thing here. Thank you for being patient. All right, I think I can share this blank Word document. All right, well, we'll just do this. So I'm going to open a new one. There we go. Perfect. All right, can you all see this blank Google Doc now? I sure hope so. Can you see it, Jen? Nope, you can't. You're still looking at something else. All right, try it one more time. There we go. Okay, now you can see it. Yes? All right, so from here, I just want to show you some quick examples. So if a DNA sequence is something like ATC, GGA, right? We know it's DNA just by looking at it because it has a thymine, right? There's a T. And in RNA molecules, there are no Ts. The thymines in RNA are replaced with uracil, so there'd be U's there instead. Now, DNA is a double-stranded animal, right? So let's clarify here. This will be 3' end here and 5' end over here. But we know that DNA is complementary, right? So the opposite side would start with 5', and then what would bind with this A? Right, a T, and then A, G, and then this would be CCT, right? 3'. Okay, good. And I'm going to slide this screen over here so I can see the chat while I'm working. Okay, perfect. Okay. But we also know that when we need to access a gene, that DNA kind of unzips, it unwinds. And so it's going to become single-stranded just at the part where the gene that we need to produce the protein that it codes for, where that gene is, it will unzip. And so we can access just the 5' to 3' strand. So we always read DNA from 5' to 3'. So this is the part that we work with here. So when mRNA makes its complementary strand to this, it's going to be complementary to the 5' to 3' strand. Maybe I'll highlight that just for fun because that's the important one. Good. So then the mRNA molecule that would be complementary to this that will form would be AUC GGA, right? Yes. So this is transcription. Excellent question. And yeah, if I had a way to highlight that somehow, this is transcription. So I will just make a note off to the side here. Nice to see you, Skylar. It's fun to see all your handles. All right, good. So transcription is when we go from DNA to mRNA, right? Because we don't need to make all the genes that are encoded at once by a single organism's genome, right? We just need to make a single gene product at once, a single protein. So transcription is when we go from DNA to mRNA. And now this mRNA travels where? To the ribosome, right? If I can type. All right, and at the ribosome, the mRNA molecule is going to look for a tRNA. So the mRNA is here. This is the mRNA strand. It's complementary to this 5' to 3' DNA strand up here. So we go from DNA to mRNA. So these two match up. Do you see that? Let me know, Skylar, if you want me to go over this, how we get the mRNA from the DNA one more time. All right. So once we know what the mRNA is, then we can figure out what the tRNA would be. And tRNA is the molecule that goes and gets the amino acids, right? So the complementary tRNA to this AUC codon for the mRNA would be UAG, right? And then the complement for tRNA from this mRNA would be from GGA, that would bind with CCU. Ah, good. So Skylar's asking about what do we call this 3' to 5' part of the DNA? That's the antisense or the nonsense strand of DNA. It would be like if you read a sentence in a book from right to left instead of left to right. It wouldn't really make any sense, but you could decode it and figure out the part that does make sense, right? Because it's just backwards. Good question. All right, so we have our tRNA. And then this tRNA molecule is what goes and gathers the amino acids. And I should clarify here, when we go, this is translation, is when tRNA is formed complementary to the mRNA. Good. So tRNA goes and gets the amino acids. And brings them to the ribosome. And Skylar's asking, so the tag CTT to UAC GGA is transcription, correct? And then when we go from this mRNA to the tRNA, that's translation. You're 100% right. You've got it. Good. Okay. And then as amino acids are added to the growing polypeptide strand at the ribosome, this is called elongation. So amino acids, remember, are small units. They've got nitrogen bases in there, and they've got, it's like an amino group, right? And then there's a special extra side chain is what gives each amino acid its unique properties. But as those amino acids are strung together, kind of like beads on a string, that string of amino acids is called a polypeptide, right? Poly meaning many. And so when the polypeptide is done, right? When transcription and translation have gone to completion, transcription have gone to completion, they're done. The ribosome will help fold that polypeptide strand. And the folding is really important, and that's what gives protein like its finished functional shape. So the sequence of amino acids is important, but then it also has to be folded just right to work. But that's the final product, is a functional protein. And a protein could be an enzyme like amylase or catalase or polymerase. It could also be a structural protein. It could be muscle fibers if it was in a human cell. All those kinds of things. But basically, proteins are what make cells work. All right, so let me put that down here in words for you all. So once transcription, translation, and elongation are complete, the ribosome folds and releases, I should say it folds the polypeptide. And releases it as a complete and finished protein. All right, so this is kind of the game plan. I will ask you on the exam, I may give you a tRNA and ask you what's the DNA sequence that it came from. I may give you an mRNA and ask you what amino acids does it code for. Ooh, speaking of amino acids and coding, which of these molecules, DNA, mRNA, or tRNA, which of those is the molecule that we use in that codon chart to figure out what amino acid comes next? Yeah, it's the mRNA. Good. All right. Yes. Yeah. So Skylar's asking if I'm looking for code. That means we're looking for the codon chart, right? Sometimes we talk about DNA as the genetic code, right? But it codes for proteins. So that's basically what we're getting at. If I want you to look up a amino acid, I'll use the term amino acid so you know exactly what I'm talking about. Good. All right. Any other questions about transcription, translation, amino acids, how we get to these things? Okay, then let's talk real quick about mutations. So I'm just gonna title this real quick, transcription and translation. And then we'll move on down here. So mutations. If the original DNA strand is, we'll use the same, well, I'll give a different one just so we're clear. 5 prime. Actually, we'll do it this way. 3 prime will be G, 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 A, T, C. And so this would be 5 prime on this side, right? And I'll do the complementary strand underneath. 5 prime. That would be C, 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 T, A, G, right? Because it's all DNA here, no U's. So if there's a mutation, That means that when this cell divided and it replicated its DNA, it made a mistake. And there's a number of different kinds of mutations that could happen. There could be a mutation where one base pair just gets totally removed. Like maybe we're going to lose this G and this C here. And if those base pairs get totally removed, then it's going to change the 3-letter codons that show up in the mRNA to go get the amino acids, right? So instead of being GGG and CCC here, we now have a very different codon, right? GGA is, oops, sorry, 5' to 3'. This CCT is going to code potentially for something different than CCC, right? So this is called a deletion because a base pair was deleted. It's also a frame shift mutation because it's shifted our 3-letter codon reading frame. And now everything downstream from it doesn't make sense either, right? Like we can't go get an amino acid with just 2 bases of a 3-base codon. So this is going to have a significant impact on the protein that's produced by doing transcription and translation on this DNA. Good. Different type of mutation could also be an insertion, right? So I'm going to put it back to what it was. So here's our original DNA. We could also do a deletion. So like if we were to delete, we did deletion, we could insert something, right? So what if there was an insertion here of T and A? That's also going to goof up the reading frame, right? Now our 3-letter codons are all different again, but in a slightly different way. See what I mean? So insertions and deletions cause frame shifts. You can also have a mutation that's a silent mutation. Maybe instead of switching everything up, you have a mutation that just oops. Undo. There we go. Say we have a mutation now that is just going to swap G. Maybe this is going to be a mutation that swaps just 1 here. Instead of changing the total number of bases, maybe it's just going to swap out that G C for a TA. So the total number of base pairs hasn't changed. We still have 2 3-letter codons. They'll code for amino acids. And maybe just by chance, and I don't know, I'm just making these letters up on the fly here, but what if they code for the same amino acid codon? That would be a silent mutation because it doesn't really have any impact. The DNA has changed, but the same amino acid will still be gotten. The sequence of amino acids is the same, and the protein that results will still be the same as one without the mutation. So that's called a silent mutation. Jen, is there anything else you were wondering specifically about mutations that you want to chat about? Frame shifts, and that results in usually big changes to the protein. Okay, good. Awesome. All right. Next up on my list of things to chat about is PCR and electrophoresis. So many of you asked about PCR, which of course is polymerase, which is an enzyme, right? Because it ends with ASE. chain reaction. So this is how we're able to make millions of copies of DNA from a single cell. So like when you watch CSI Las Vegas and they find a single skin cell underneath the victim's fingernails and they're able to identify the PERT from that DNA, it's because they did PCR on it. And it's incredibly powerful, super cool tool. We're able, like we've done PCR on DNA from Neanderthal remains. We've done it on, weird, like Pleistocene megafauna, like giant ground sloths that are thawing in the permafrost in the Arctic, dire wolf pups. We've been able to get DNA out of crazy things using the polymerase chain reaction. Which is super exciting for science. It's also really useful as a microbiologist because we can only grow in the lab a tiny fraction of the microbes that exist on Earth, but they all have DNA. So if we can just find them and extract their DNA, we can still study them even though we can't grow them in the lab. But for the kinds of questions I've been asking you in our biotechnology unit, our chapter, we've been using it kind of to identify disease, right? So say there's Ebola virus has broken out and we want to try and see which patients have Ebola and which, have something else that's not nearly as scary. So what you do first is you would extract the DNA from the sample. So you might take like a saliva sample or like swab their nose, someplace where you think the virus or bacteria would be replicating. And so you'd extract the DNA first. And then the next step is to do polymerase chain reaction. And what polymerase chain reaction does is it replicates how DNA is replicated in the cells, but it does it in a test tube instead. And it does it over and over and over again. So the first step in either case is to unwind the DNA, right? So if we're going to unwind the DNA in cells, we'd use the enzyme helicase, which unwinds the helix. In PCR, we use heat. So by heating it up. The bonds that hold two strands of DNA together are the weakest bonds in all of the structure of DNA. So if we heat the DNA up, the first part to break will be the bonds that hold the two strands together. So double-stranded DNA will break apart into single strands. So that's the first step. Next, we need to apply the primers. Step 2, primers. So in DNA replication in a cell, there will be primers, RNA primers that will bind, and that gives the adding new bases a place to start. So we actually supply